Today, I'm thrilled to share our work on a novel mechanism by which the classic anti-leukemic drug Homoharringtonine, HHT, suppresses acute myeloid leukemia, AML. Despite the promising anti-AML efficacy of HHT, a CML drug, and its synergies, its molecular mechanism and direct cellular targets remain elusive, limiting therapeutic optimization. EWSR1 is established as the direct target of HHT, where drug binding triggers pathologic phase separation and YTHDF2 entrapment, thereby serving as both effector and predictive biomarker in AML. Proteome microarrays and cellular validations identified EWSR1 as the primary HHT target, whose elevated expression correlates with poor AML prognosis, establishing its dual role as a therapeutic target and prognostic biomarker. Elevated EWSR1 expression, particularly in leukemic progenitors, directly predicts effective HHT response in AML. Multiple orthogonal approaches confirmed a direct, high-affinity interaction between HHT and the C-terminal RRM of EWSR1, validating it as a bona fide cellular target. Photoprobe mapping and mutational analysis revealed that HHT binds the EWSR1 RRM domain via critical hydrogen bonds with residues T393, P396, and I400, defining its molecular mechanism. EWSR1 undergoes LLPS via its N/C-terminal disordered regions. GFP EWSR1 formed dynamic, liquid-like condensates in cells, as evidenced by FRAP and fusion, properties that were disrupted by 1,6-hexanediol, confirming its phase-separating behavior in relevant models. Both the N and C-terminal domains of EWSR1 independently drive phase separation. HHT specifically enhances the condensation of the C-terminal domain and reduces its fluidity, indicating a stabilizing liquid-to-solid transition. Biopsy and immunoblots confirmed that HHT promotes EWSR1 self-interaction and oligomerization. Complementary structural analyses revealed HHT induces unfolding of the C-terminal RNA-binding domain, driving condensation via allosteric oligomerization. Quantitative proteomics identified YTHDF2 as a principal EWSR1 interactor during phase separation. Their strong co-expression in AML progenitors, amplified by HHT via the YTH domain, reveals a critical EWSR1 YTHDF2 axis as a promising therapeutic target in AML. HHT reinforces the direct binding between the C-terminus of EWSR1 and the YTH domain of YTHDF2, leading to YTHDF2 sequestration into condensates and impairment of its mRNA binding and decay function. Transcriptome analysis revealed that HHT treatment and EWSR1 knockdown produce near-identical gene expression profiles, primarily affecting RNA processing and immune pathways. This striking congruence establishes EWSR1 as the central functional target through which HHT disrupts oncogenic RNA networks in AML. HHT increases MA modification on apoptosis-linked mRNAs, impairs their YTHDF2-dependent decay, and ultimately promotes leukemic cell death through RNA regulation. Using AML mouse models, we found that HHT's ability to suppress leukemia was severely compromised upon EWSR1 knockdown, proving EWSR1 is indispensable for its therapeutic effect. The essential role of EWSR1 was further corroborated by survival analysis. HHT extended lifespan in wild-type mice but not in EWSR1 knockdown hosts. Consistently, in the Kasumi-1 model, combining HHT with EWSR1 depletion offered no additive benefit, confirming EWSR1 as the critical target. Our study converges on four pivotal conclusions. EWSR1 is the direct target, explaining HHT's anti-leukemic mechanism. It triggers allosteric phase separation, driving EWSR1 condensation. This process sequesters YTHDF2, disrupting mRNA decay. Critically, EWSR1 is essential for efficacy and a predictive biomarker, bridging molecule and clinic.